Center for Misophonia and Emotion Regulation webinar series. This is our end of summer/fall event. We do 3 to 4 of these per year, and we're excited today to have Dr. Julia Campbell, who is an audiologist from the University of Texas, Austin. Dr. Campbell is going to share with us what audiologists do in treatment for misophonia and what audiologists have helped to uncover about what's happening in misophonia in terms of hearing processes and brain processes related to misophonia. So we're going to get there in a few minutes, but first things first, while people are trickling in, welcome, welcome. Again, I'm Zach Rosenthal, and if you're your first time here, we always take a couple of minutes up front and welcome people and orient them. Here, if you are interested, as you're listening, in going to our website to learn more about what we do at Duke at the center, misophonia.duke.edu is our website. If you are an Instagram person, you can follow us at Duke Center for Misophonia. And if you enjoy watching YouTube videos, short and long, we are always adding to our YouTube channel, Misophonia series at Duke CMER, CMER. You also can email me anytime if anybody has questions or ideas or things that you want to share. My door is always open, and I'm happy to communicate with you. We have a big team here at Duke, and we're really grateful for the way everybody here helps us with everything we do. We do research, we do treatment, we provide education, as we are today, to the lay public and to clinicians. And we also provide advocacy and help raising awareness for misophonia. So thank you to my team, to everybody who contributes to this. Also, big thank you to our partners for this webinar series, who have been our longtime partners. Dr. Jennifer Brout, who leads the Misophonia Research Network, and Cris Edwards, who is the founder and director of SoQuiet.org, which is a fabulous nonprofit advocacy group for misophonia. And Cris will be here with us today to help facilitate the moderation in the Q&A portion of the webinar. Dr. Brout sends her regards. She unfortunately is not able to be with us today, but she will be back next time. Thanks again to my team, but in addition to my team, I really want to thank all of the anonymous families who have generously supported philanthropically the work that we do here. All of the things we do to help change the world related to misophonia, literally every single thing we do, it can't be done without the generosity and support of the various families, whether they're providing small amounts or big amounts. We just really want to thank you, all of you, for your support of this. And thanks to all the advocates who support research, education, and treatment of misophonia everywhere, all of you. This webinar will have clinicians, researchers, it will have other people interested in supporting misophonia. Most importantly, it will have people who are suffering as sufferers themselves of misophonia, and parents and loved ones, as I personally am a loved one of someone with misophonia. We also will have many of those people on today. And so just thank you to everybody. Thanks to everybody for everything you do to help raise the tide here for all things misophonia. Brief announcement, excited. I know this is a bit cryptic, and it is what it is, but tomorrow morning, Eastern time, we're going to be releasing a video through our center with a recognizable person who is going to be publicly sharing that they have misophonia and asking for everyone's help to raise awareness. So there's a little bit of a teaser. We're really excited. We've been working with this person for over a year to make this video, and check out our website, our Instagram, or YouTube if you are curious and want to help raise awareness. You can spread this video, and the idea is we really want to ultimately get things to a place in this country and elsewhere where when you go to see a doctor or a clinical provider and you mention the word misophonia or you present with those symptoms, that the doctor or clinical provider knows what you're talking about. That's our end goal, and that's part of why we've made this video for tomorrow morning. So check it out. Okay, so today's webinar has a couple of parts. We're in the first part. We're about to wrap it up with me just welcoming and orienting everybody. And in just a second, we're going to shift to Dr. Campbell. And we will go till about 35, 40 minutes after the hour. And we'll aim to have a good stop no later than 40 minutes after the hour. We will stop recording at that point, and then we will take Q&A, which Cris Edwards from So Quiet, Lisa Kelly from my team, and I will also help with, in addition to, of course, the fabulous Dr. Campbell, who will answer all questions related to her talk. That is our plan for today. I'm going to just introduce Dr. Campbell here and then turn it over to her to share with you her slides. Dr. Campbell received an AUD, as a doctorate in audiology, in 2009 from the Department of Speech, Language and Hearing Sciences at the University of Colorado Boulder. In 2015, she made us all feel quite unworthy by receiving a combined triple, that's 3, PhD, a triple PhD in speech, language and hearing sciences, behavioral neuroscience, and cognitive neuroscience, also at University of Colorado Boulder. She is the PI, principal investigator of the Central Sensory Processes Laboratory, and her work in general focuses on the plasticity of sensory processing in the brain in both typical and clinical populations. She also, I will just say as an aside, Dr. Campbell is a colleague of mine and ours, and is someone who leads the Misophonia Research Fund Scientific Advisory Board, which is how we know each other. So she is very plugged into the science of misophonia, and we're all excited to hear what you have to say, Julia. So welcome, and please feel free to share your slides and take it away.
Great. Thank you for that very kind introduction. I'll go ahead and share. Okay, welcome everyone. I'm Dr. Campbell. I'm going to be talking about audiologic assessment in misophonia. So there's two parts to this presentation. I'm going to first try to orient everyone to what is an audiologist, what do they do? If you go to an audiology appointment, what tests might you undergo? What do those mean? Do those have any tie to misophonia findings in the literature? And then the second part of the talk is going to focus on comorbid auditory disorders with misophonia. So trying to separate those and identify what specific issues are going on in an individual. And you'll get to see some of the controversy, the beef within the field. So that'll be fun. Okay, so what is an audiologist? This is an individual with a post-baccalaureate degree. And most recently, in the last 10 years, they've changed that from a master's to a doctorate. So you'll see individuals who are audiologists have an AUD behind their name. They are trained to perform screening and diagnostic hearing assessments, ranging from the outer ear, middle ear, inner ear, and central auditory nervous system. And they are also trained to perform rehabilitative interventions. So things like fitting hearing aids or performing counseling, different managements for disorders like misophonia, tinnitus, hyperacusis. So we'll just review very quickly what the auditory system looks like. We have the outer ear here, which also consists of the external auditory canal. This is where all our wax gets generated and you might get plugged up. Here's the middle ear. So we have the eardrum right here. And then we have the 3 smallest bones in the human body that conduct sound. And this is an air-filled cavity, but there's a mucus lining within that cavity. So it tends to be a bit damp in there. And this connects directly to the inner ear, or what we call the cochlea. It looks like a little snail, and the balance system, which are these semicircular canals right here. And in the cochlea, we go from an air-filled system in the middle ear to a fluid-filled system. And we have little hair cells within here that respond to the fluid movement bending, and they send electrical signals to the auditory nerve. Those electrical signals travel to the brainstem, and we hit some important hubs in the brainstem, like the cochlear nucleus, the superior olivary complex, up to the inferior colliculus, where finally we get to the brain and to primary auditory cortex, which is on the temporal lobes on both sides of the head. And from there, we just go all over the brain to frontal cortex, motor cortex, things like that. So these are the basic types of tests you'll see conducted during an audiology appointment. We're going to have behavioral audiometry and speech perception testing, middle ear function, inner ear function, the auditory brainstem response, and then cortical auditory evoked potentials. So we're literally moving from the peripheral part of the auditory system to the central part. So what might you expect in audiometry testing? This is our lovely beep task, where I'm sure many of you have had this. You go into a sound booth, you hear some beeps, you raise your hand or you press a button to say whether or not you heard that beep. And what we're looking for when we do that is threshold of hearing. And that means the point that you can hear that sound 50% of the time. So that's your threshold for hearing. And this allows us to assess the presence of hearing loss, but it can also alert us to something called hyperacusis, which we'll talk about a little bit more later in the talk. And this can be generally defined as a hypersensitivity to the level of sound. So not necessarily the type of sound, but how loud it is. And then speech perception, where we might read single words or you might listen to sentences and background noise, also gives us information on how well you're functioning in the everyday world, because we don't have a beep-based language. We listen to sentences, we have background noise constantly, we're never in a quiet environment. So this is very important testing to have done. Okay, so some of you may have seen this before too. This is an audiogram, and this is where we basically graph the results of your hearing test. So here on this horizontal axis, we have frequency, or you can think of it as pitch. And this is very low frequencies, and it goes to higher frequencies. And then this is the intensity of the sound, defined in decibels, and very quiet. I know you might say, well, 0 would be no sound, but on this scale, 0 is still some sound energy, ranging all the way up to what you might hear when a jet passes right over your head. So very, very loud. Normal hearing, and this is now controversial in our field as to what that really is considered to be hearing thresholds at 20 decibels or below. So that's the range of normal hearing. Anything outside or above that is considered hearing loss. And there's varying degrees of hearing loss. There can be mild hearing loss in this range, moderate in this range, and then severe and profound. Profound hearing loss is what we mean when we say deaf. There's no sound being perceived by the brain, all right? I would have to say, though, that when hearing loss is present, it's usually not equal across frequencies. So typically we'll see better hearing at different frequencies and worse hearing at other frequencies. Well, you might say, why do we only test these specific frequencies? We know that the range of hearing extends up to 20,000 hertz. So why aren't we testing these very, very high frequencies? In our field, we call them extended high frequencies. And there's two reasons for that. One reason is that the speech frequencies, the frequencies most heavily weighted in our perception of speech, are in this, I would say, 1,000 to 4,000 hertz range. So we want to focus on those because those are the ones that we're using every day. But also, the hearing aid companies typically do not amplify frequencies above 8,000 hertz. And even amplification of frequencies at 8,000 hertz is not great. So there's also, I would say, a corporate reason for only testing to a certain range. But I'm happy to say that the field, and especially the clinical part of the field, is moving to now test these extended high frequencies. And that may be very important in some of these different auditory disorders. Unfortunately, there's no normative data for what typical hearing should look like at these frequencies. So that is a problem and something that needs to be addressed research-wise. But I will say, we did a study where we had young college students with typical hearing across the board and very, very minimal tinnitus. And tinnitus is a disorder where one perceives sound when there's no presentation of sound in the environment. So it usually is a ringing, it can be static, it can sound like different things. And we found that these students had atypically good hearing in these extended high frequencies. So that raised the question of, can your hearing be too good? Can that cause issues? And we actually looked at this in a preliminary study in relationship to misophonia, and we didn't see that same relationship, which might indicate, and you'll see this throughout the talk, that misophonia is more of a central issue than a peripheral auditory issue. Let's move on to the middle ear. This is one of my favorites. So for middle ear function, you'll undergo something called tympanometry and acoustic reflex testing. Tympanometry is when we insert a probe into the external canal and we present a tone or a beep. And while we're playing that beep, we change pressure. And we're trying to push the eardrum in to the middle ear space and then pull it back out. And while we're changing pressure, we're measuring how much energy of that sound or tone passes through the middle ear system. And we get these little mountains back, these little images. So this is a tympanogram, and this indicates normal passage of energy while we're changing pressure. If we get a flat tympanogram, that usually indicates that there's fluid, like when someone has an ear infection, or something is blocking the sound from getting to the inner ear. And then you can have also different types of tympanograms. For example, when the most sound is passed during negative pressure, and that can indicate, eustachian tube dysfunction. We also test the response of the middle ear muscles to loud sound. And this is actually a very interesting phenomenon and something that I've recently learned a bit about. But there are some middle ear disorders that could relate to misophonia. We just don't understand yet whether that is true. And those are called the tensor tympani syndrome and acoustic startle disorder, or acoustic shock disorder. And these are when the muscles of the middle ear are not functioning correctly, and they might contract too much, and it can cause pain. It can cause a feeling of fullness in the middle ear, and it can also lead to fear of sound, which can be an attribute in misophonia. All right, next we can test inner ear function. So this is where we look at the function of those hair cells in the cochlea. These are called otoacoustic emissions, or OAEs. It's a super fun test. We do it a lot in babies to see how their inner ear is functioning. We put a probe again in the ear, and we just play a range of sounds across different frequencies. And this is crazy, but when you present a sound to the ear, if those hair cells are working correctly, they play a sound right back out that we can measure. And so some people think of them as an echo almost from the inner ear. And that lets us know that they're working. And this is a little graph just showing that indeed we are able to record responses from those hair cells. Auditory brainstem response. So this is actually how we do test newborn hearing. We screen them, and it's a federal law that this must be done. If parents don't want this done, they sign a waiver. And this allows us to look at the response of the eighth nerve, the auditory nerve, all the way up through the brainstem. These different peaks represent those different hubs in the brainstem that respond to sound. And we can see is the integrity of those central pathways appropriate for that baby's age. And we can use this in adults to see if we get atypical central processing in the brainstem or if it's higher up. And there's only been, to my knowledge, one study looking at this in misophonia, and they didn't find any atypical auditory brainstem function in that study. So with all these tests, these are something that you might see as you are a patient or a professional health provider who's working with an audiologist. And what we're really trying to do as audiologists is a differential diagnosis. We're trying to rule out hearing loss because that can be treated with amplification. We're trying to rule out medical or ENT-based disorders that should be addressed first, things like that. We can't diagnose misophonia using these measures because there is not enough data. There appears to be no relationship with auditory thresholds and misophonia. There doesn't appear to be a relationship with OAEs, or inner hair cell function. And the middle ear, like I said, there seems to be some subtype disorders like acoustic shock disorder, tensor tympani syndrome, that could relate or trigger an onset of misophonia themselves. What we can do is work with a multidisciplinary team and diagnose misophonia, again, by ruling out these other disorders and utilizing multiple questionnaires to provide a holistic view of what the patient is going through. And then according to those questionnaires, implement a treatment plan. So that's what we can do currently in the field. Research-wise, we're hoping that we can find a relationship of some sort with these diagnostic tests and misophonia characteristics, but that just hasn't been done yet, unfortunately. So some auditory disorders that we are also going to try to rule out if a patient complains of some misophonic symptoms, the big two are going to be tinnitus and hyperacusis. And it appears that there's a very high level of comorbidity with these two disorders and misophonia. Again, there hasn't been a great study that's defined the prevalence of this comorbidity, but one study suggested that patients who have tinnitus and or hyperacusis, 42% of them may have misophonia. So it's very high. We want to parse this out so we know how to proceed with intervention. So hyperacusis, again, I'm going to redefine it. We don't have a consensus definition like we do for misophonia. So this is problematic in the field and for healthcare providers, and for patients. It's generally defined as a loudness intolerance disorder or sensitivity to the intensity of everyday sounds. Again, more about the intensity or the level of the sound than a specific type. But you can see how these could overlap. This is just an illustration of what hyperacusis might look like. This is that audiogram that I showed you earlier, but flipped upside down. So now we have quiet sound here at the bottom, going up to very, very loud. And then we have our low frequencies all the way up to our very high frequencies. So threshold of hearing is shown here. This is a typical threshold of hearing. We tend to hear better in those mid-speech-based frequencies. Okay, so speech and music tend to occur in this decibel range. The threshold of pain or feeling sound is up here at very loud levels. A typical person with normal hearing will have an uncomfortable loudness level, the point where sound starts to become uncomfortably loud. They don't want to listen to it, around 100 decibels. In hyperacusis, this uncomfortable loudness level is dropped into the area of speech. So it can become very, very problematic and affect everyday function. Now, in audiology, the beef with hyperacusis and misophonia is, are they separate disorders, or is misophonia a type of hyperacusis? And that's been the argument that's kind of ongoing. So one paper proposed that hyperacusis consists of 4 main categories, and that's loudness hyperacusis, which is what I pretty much just defined to you, pain, annoyance, and fear. All right. I think we could agree that misophonia might best fall under the annoyance category of hyperacusis, but it could also fall under fear. And we also call this phonophobia, fear of sound. So some people, myself included, have said that misophonia should be considered a separate disorder from hyperacusis. Others strongly disagree. So are these the same or different? Well, we don't have any research to really disentangle the two. But it's my professional opinion that it's likely both. If measures show that hyperacusis and misophonia are present together, it's possible or likely that misophonia is indeed a subcategory of hyperacusis. But if measures are only showing traits for misophonia and not hyperacusis, then we have an independent standalone disorder. And I'll show you how we can disentangle these a little bit more. So this is the general feeling currently in the field that you can have hyperacusis without misophonia. You can have misophonia without hyperacusis, but you can also have both. And then it becomes a little more trickier to treat. But it is very important to do the appropriate testing to determine if this is indeed the case. So the clinical approach now in today's time should always consist, if somebody is complaining of a sound tolerance disorder, sensitivity to sound, levels of sound, types of sound, these individuals should always have an audiologic assessment, excuse me, and a psychologic assessment. These are going to go hand in hand, and the providers should be communicating with each other in a team format. The audiologic assessment is going to consist of what I just showed you, those various tests. We're also going to be using multiple questionnaires. One questionnaire should not be used because they're all different and they all provide a multifaceted view of that patient and what they're going through. So there was a really great study recently done by Kula and colleagues, and they actually reviewed all the current questionnaires for misophonia and sound tolerance disorders to see which fit a very stringent criteria for being appropriate clinically. And they came up with a table. So I would encourage you to look at that paper if you're a provider. Unfortunately, some really great questionnaires were published right after that study. So there's that. Hyperacusis questionnaires, we need to have more than one. Some really great hyperacusis questionnaires are the hyperacusis questionnaire, not a really inventive name, the hyperacusis impact questionnaire, and the IHS. And then for misophonia, we have many questionnaires. I think the strongest ones are the misophonia questionnaire, the AMISO-S, and the DMQ, the Duke Misophonia questionnaire. However, there is a new questionnaire that I think might trump a lot of these, and I've provided the link. It's called the MIST-A, and it was created by a group at Vanderbilt, and it's phenomenal. I wish I could click out of there and show you, but it covers all the categories of hyperacusis and misophonia. And then once the patient has filled out the questionnaire, it calculates a percentage of each category and what that patient is experiencing. So they might have a hyperacusis dominant presentation, or they might have a misophonic dominant presentation. It's really, really good. I strongly recommend you look at it. However, it has not been validated, so it still needs to go through some stringent reviews. Audiometry, surprisingly, can also help us look at hyperacusis. It's almost the reverse of what we were doing earlier when we were trying to assess threshold, which is very soft. Now we're going to try to assess the uncomfortable loudness level for these individuals and see if that's around 100 or lower. And if it's lower, then we have a red flag for hyperacusis. However, the issue with this measure is that it doesn't correlate very well. It doesn't agree very strongly with hyperacusis questionnaires. So then, do we rely on this uncomfortable loudness level measure, or do we rely on a questionnaire, or do both need to be positive for misophonia, or excuse me, hyperacusis? It would help to have a consensus definition to answer that. Extended high frequency thresholds, like I mentioned before, more and more clinicians are starting to do. Could these be too good? Could there be subclinical damage in this area of hearing that is related to misophonia? We don't know yet. Our pilot study showed that there didn't appear to be a relationship, but it was a very small study. So this is something to look at as well. And then otoacoustic emission testing, that inner hair cell test for the inner ear. Excuse me, it's an outer hair cell test for the inner ear. That can indicate subclinical damage. So it is possible to have on that audiogram normal hearing, but the hair cells in the inner ear are still damaged because this measure is more sensitive than the audiogram to physical damage. So this is something any good audiologist should do. Even if your hearing test is normal and everything looks good, they should do otoacoustic emissions to determine if there might be slight damage to the inner ear and what that could mean for the patient. So to summarize, future for best practice in clinical care, we really need to answer the question of, is hyperacusis and misophonia the same or different? And this is really important in educating other healthcare providers and identifying targeted intervention that is appropriate for that patient. We also need to come up with some more sensitive testing for the auditory system that might relate to misophonia. Again, we don't hear in beeps. That's not how we function, so it's not a great measure. A group in France came up with a really great environmental sounds test that might be able to differentiate between hyperacusis and misophonia. So that might be where we're headed, and I would love to use that in the clinic. And then we might want to start looking more at the cortex, the brain, the central area where we think this issue is actually occurring. And that could be cortical auditory evoked potentials, which is a setup just like the auditory brainstem response. Instead, we're looking at the higher part of the brain, the cortex. And that could be used clinically quite easily. People don't need to be trained in how to do that because the ABR test is so similar. So this would be ideal. And one study did show that there was a change in cortical potentials in response to sound. It hasn't been replicated, and there haven't been many more studies looking at this in misophonia. So to summarize, audiology needs to pick up the pace and really step to the plate and start conducting some research studies that indicate what tests might be sensitive to misophonia. And I think we're ruling out that the periphery is involved. It's going to be a central measure that's going to help us. All right, I hope this was helpful. And that's all I have.
Thank you, Dr. Campbell. Thank you so much for that. I really appreciate, as a psychologist, you walking us all through the real kind of basics of sound and sound processing. I really, really appreciate that. I'm sure other people do as well. And then raising this really important question about hyperacusis and misophonia and which is which and how do we even think about that? And then especially really appreciate for all of the people here who are listening who may go seek out an audiologist. Really appreciate you kind of saying these are the tests that we would recommend.
Mm-hmm. Yes.
And for those of you who don't know, and Julia, maybe you can unshare for here if you don't mind.
Of course.
I'll share your screen. Thank you. So for those of you who don't know Dr. Campbell and her work, she is quite literally the person who recently published the recommended guidelines for audiologists to use worldwide when assessing misophonia. So we really appreciate you as the person who literally wrote those suggested guidelines here with us today, talking about what you think those things should be. And my hope for everybody who's here, who is a loved one or who is a sufferer, is that you can take Dr. Campbell's wisdom, take her suggestions, and find it useful as you go to your audiologist. And so